So, welcome to the second in this machine learning seminar series that I'm doing, looking at different examples in biological sciences. So, 2 weeks ago now, we had a look at linear regression with regularization, looking at epigenetic clocks as an example. Today we're going to have a look again at quantitative prediction, but this time using support vector machines and using this example for looking at predicting promoter strength. And then we'll cover some different considerations and outputs for different machine learning models in the final three as we go through. So, what I will cover, for those of you who came to the last one, this will look very familiar. I'll start off with a quick introduction to machine learning, then move on to introducing the paper, so what data sets they've used and what they were aiming to do, then look at the pre-processing that they've done, looking at kind of feature extraction and things like that. Then I'll go through how the model works in a sort of conceptual way, and how they did it in their paper, then how they validated it and how well it worked. And towards the end, we'll go into tidy models and have a go at running a similar model ourselves. And finally, just wrap up with some further exploration of what they did with their model after they'd built it. So, what is machine learning? I will run through this relatively quickly because probably for the most part people will watch all of these, but just in case, we, on the whole, in a usual data analysis workflow, we'll start off with collecting our data. We will then do some kind of preparation and formalize our result using some kind of test to give us an outcome of interest. So we'll start off with some kind of raw data. We will visualize and explore it to make sure we understand what's happening. We'll probably filter and clean our data in some way and might loop that round a few times until we understand our data and are happy with it. Then usually we'll do some kind of statistics to formalize our result, which will give us some outcome that we can report. So that there is or is not some statistically significant result is usually what we would end up with. If we are using machine learning, instead of doing statistics, we will do modeling. Sometimes that can be what we report. So it might just be that we are interested in showing that our predictors of interest can predict whatever our outcome is. And so that might be what we report. But for the most part, with modeling, we want to make predictions. So we want to make predictions where we don't necessarily know the outcome, so we can sort of use it in this kind of setting. So the steps in machine learning, firstly, we obviously have to generate the data for samples where the outcome is known. So we need to know the outcome in as large a data set as we can get our hands on, essentially, is often the case. But we also need it to be high quality enough. We've all heard of rubbish in, rubbish out. That is very much the case for machine learning. We then pick a modeling method and build a model based on the kind of data that we have and the kind of outcome that we want. And we can evaluate our model and refine the model in different ways to then make predictions where the outcome is not known. And we can also then test those predictions and see how useful our model is. There are some key differences between models. The first one is the outcome type, so whether or not you want to make quantitative or categorical predictions. So if you want quantitative, you will do a regression. If you have categorical, you will do a classification. Some model types can do both, some can only do one or the other. And the other difference is the input type of data that we have. So some models require all their variables to be numeric. So that may mean that we have to convert any categorical values to numbers, which we will see today. There's also sometimes expected behavior of our input data. So some models need our data to be normally distributed or on a similar kind of scale. And there's also variation in the sort of number of viable measures or the sort of data set size that you might need for different model types. So some work well with less data, some work well with more data, essentially. These are some of the different kinds of models. We've already looked at linear regression last time. This time we're going to have a look at support vector machines, and we will look at a couple of the others as we go through these sessions. So the paper for today is this one, Construction of Precise Support Vector Machine-Based Models for Predicting Promoter Strength. As I said in the last one, this is not a journal club. I'm not necessarily going through all of the details of this paper and critiquing it in any way. I'm just using it as a basis, as an example of what we can do with machine learning and some of these kind of considerations. So basically the aim of this paper was to build a model to predict promoter strength, as you might expect from the title. Essentially with sort of thinking about applications in sort of synthetic biology predominantly. So you could potentially predict or control the yield of a particular protein that you want to produce by changing the promoter sequence potentially. So the data set that they have is a mutation library of E. coli TRC promoters. So they have 100 different promoter sequences with different mutations within them and some kind of corresponding strength value for the strength of the promoter. The length of the promoter is 224 bases in all of these cases, and it was generated by error-prone PCR based on the wild-type promoter. So one note with this is it only produced or introduced a sort of less than 30% mutation rate. So actually for a lot of them, all of the bases are for the most part the same. And also, yeah, it just slightly questions the sort of generalizability of this, but just as a note there to be aware of. The promoter strength was measured by detecting GFP expression in E. coli using flow cytometry, and then the strength is relative to the wild type sequence. So the first thing they did with this is to convert the categorical or sequence data to a dummy variable. So because some models can't use categorical data as an input, of which SVMs are one of those, we have to create something called a dummy variable. So we're essentially encoding our information into numbers, which is usually binary, so you'll either become noughts and ones in your data. In this paper, they converted the original sequence data to a digital matrix for an SVM model input. So all of the A's became 1000, or the G's became 0100, C's became 0010, etc. So if you had AGT as a sequence, it would become this series of ones and zeros here. And this is something called one-hot encoding, where essentially for each of your possible categories, or in this case bases, you have a different sequence of zeros and ones. So you end up with a number of columns, or in this case rows, equal to the number of possible values within each base. Another way that you often see this done, which was not done in the paper, is to have some kind of baseline value. So one of your categories, or in this case, one of our bases would just be all zeros. So in this case, you would only have 3 rows or columns rather than 4, and A would always be 0, G is 1, 0, 0, etc. So they're just slightly different ways of doing the same thing, just to be aware of. One problem with, well, both of these approaches, but particularly this one, is you make a really sparse data set. So you end up with an awful lot of zeros across your data set, which again, something to be aware of at this point. So once they'd done this, they split their data to be 90% training and 10% testing. So they could train and fit their model on 90% of their sequences and then see how well it performed in the 10%. So for the model that they used, their input for the training data was 90 different promoter sequences that have been converted to numeric values. Their output that they wanted was the predicted promoter strength, which is obviously a quantitative output. So we need some kind of regression model. And because we've converted to numeric predictors, we don't have that as a sort of issue when we're trying to think about which model to use. So the model they used for this is something called the radial basis function, or RBF, support vector machine, SVM. So what is that? We'll start off with simple support vector machines to see about that. So essentially support vector machines at the simplest level that we can think about to build up, divides groups based on a particular threshold. So if you imagine we have group A, group B here, we could put a line here and call that our threshold. Anything that's below the threshold becomes group 1, anything that's above becomes group 2. The thing to note with this is these 2 points are therefore super important. We're essentially just choosing the midpoint between these two. And so these become something called our support vectors. So these are the sort of key values that determine where our threshold ends up. And they are essentially at the edge of the clusters of our groups in this example. We then have something in the middle between these two support vectors, and this is called our margin here. And when we are trying to decide where the threshold is, we want to minimize our margin. Maximize our margin so that we have the biggest difference between our two groups. That works well if we have nice separation between our groups like this, but if our training data also included this data point here, then it would mean that our threshold would be somewhere around here instead. And if in our testing data, say, we had this data point, it would now be categorized as being group 1 rather than group 2, even though it looks sort of visually like it might be closer to group 2 than group 1. So what we do to get around this is allow for misclassification in the training data to reduce overfitting. So what we can do is generate a threshold based on multiple runs with different subsets of our data to try and optimize the position of this threshold. And we can do this using cross-validation, which I briefly went through last time. But as a reminder, we split our data into a certain number of chunks, essentially. So in this case, it's 10-fold cross-validation. So we split our data into 10 different parts, and we train a model on 9 parts of that and test it on 1 part until we have trained on the same piece of data 9 times and tested on it once. So when we do that, it can allow for misclassification of the training data to reduce overfitting. So in this case, we might then get back to our original threshold here with these 2 data points being our support vectors again. We then end up with something called a soft margin rather than just a margin, where we might have data points in between our support vectors over here. So we might have ones that are within the soft margin and classified correctly, but we also might have some misclassification within our soft margins as well. And one of the parameters that we can choose when we are defining our model allows us to balance the number of correct and incorrect classifications within these kind of soft margins here. So that helps us balance between overfitting and essentially underfitting for our training data. So that's all an example if we just have one value, but obviously we have many predictors when we do machine learning. So just to say, if we have 2 different predictors, then our threshold will instead be a line rather than just a single point. And again, it might be that that line or that threshold becomes this rather than the original there, with potentially some misclassifications. if using cross-validation, we find that that's a better fit overall for our data. So that's all well and good, but sometimes we have more complicated data to try and deconvolute. So here we have, again, our two groups, but this time they're not nicely separated on this predictor here. So this is where support vector machines get slightly cleverer. So what we do is we project our data into another dimension. So we're going to add another dimension here, and we can square all of our values. So this is just the original value plotted against the squared value. And now we can draw a nice line to separate our data again. So support vector machines essentially project our data into higher dimensions and then find what's called a support vector classifier. So that's essentially finding our threshold to try to divide between our groups. We can use or define different kernel functions, which differ in how they transform our data to set that kind of threshold. I've put transform in sort of quotes here because it doesn't actually behind the scenes transform the data. It just defines the threshold in a different way because it's less computationally intense. But essentially, when you're thinking about it, you can think about it as transforming the data. So there are sort of three common kernel functions that are available within tidy models and the engine that I'm going to show you later. So the first one is linear. So this maximizes the width of the margin between the classes using a linear class boundary. That's essentially what I was showing you initially. Obviously, for all of these examples, I've shown classification because it's sort of easier to visualize in these ways. We are actually using it for regression, which is slightly different because we want to predict quantitative value, but the sort of concepts are essentially similar, but you're essentially using like a linear fit rather than a linear like threshold to predict your quantitative value. The second type is polynomial, and you can have different degrees of polynomial kernels. So if we have, we set our d, which is our parameter for our model, as 2, which is our degree of polynomial, then it will compare the original data with the squared data between our points, which is essentially what we looked at the last slide of the example. If you set d as 3, then it will compare original squared and cubed across 3 dimensions. And so our support vector classifier that time would be a plane rather than just a line. And if we have d of greater than 3, then we can compare in more than 3 dimensions, which is not easy to visualize. And we end up with a support vector classifier that is a hyperplane across more than 3 dimensions. This can also be optimized using cross-validation, so we can figure out from our data what the best degree of polynomialism is. And the final kernel function that I'm going to talk about is radial basis function, which is what they used in this paper, which finds support vector classifiers in infinite dimensions, which obviously is impossible to visualize, but we can essentially think of this as when you have nearby values, they will have more impact on the prediction than further away values. So it sort of acts similarly to a weighted nearest neighbor model. And again, you have a tuning classifier or a tuning parameter, sorry, in your model where you can tune the weighting. So you can say sort of how much impact each data point has on other data points that are nearby versus further away, essentially. So in the paper, they actually tested several different kernel functions and found RBF or radial basis function to work the best. So they went with that. They also tested different sizes of test versus training data from 10 to 90 in the training data and found that with more training data, they got better predictions. So used this 90-10 split. This is what this is showing here. So at small training sets, it wasn't very good, and it was much better when you had larger training sets. For this, they used mean squared error and R squared to evaluate the performance. We will come back to those in a bit, but essentially mean squared error looks at the average of the squared residuals, so the sort of average of the squared errors of the model. So you want that to be smaller. And R squared looks at essentially the amount of variance in your predicted value that is explained in your sort of original value. So essentially you want that to be larger and it's better. They use something called SVM Toolbox in MATLAB. I am not familiar with MATLAB myself, so I will not go into any more details on those, but we will see how it looks in tidy models later. So as I said, there are a few different parameters that we can set for this kind of model type. There are in fact three. The first one is the cost function, which in the paper they set at 128. This is essentially the cost of predicting within or on the wrong side of the margin. So if you remember, we had the soft margin between our two support vectors. So if data points fall within those, what's the kind of cost for that? So if you have a larger value, your model will be more fitted to the data it sees. If you have a smaller value, it will be more generalized. So larger value has the potential to overfit, smaller value has the potential to miss some complexities. We then have something called sigma for the radial basis function. This is exclusive, as you might expect, to the radial basis kernel. So you don't need this for linear or polynomial ones. And that's because it's essentially coming back to a similar concept as the nearest neighbors that I was talking about before. So how far the influence of a single example reaches. So if you have a smaller value, then it will be more fit to the data. If it's a larger value, it's more generalized. The final one is called epsilon. This is the insensitive loss function. This is only defined in regression and not classification because it's essentially a region around the predicted value where errors are not penalized. So in this case, it was set at 0.01. So if your predicted value is less than 0.01 away from the actual value, it won't be counted as an error, essentially. It's close enough to your actual value to be considered correct. And again, if you make this larger, you're going to accept more error. If you make it smaller, it's going to be more precise and therefore more fitted to your training data. Also, all of these, they figured out using tuning of the model and cross-validation to find good values for these based on their approach. So they ran all of this and got some results. They ran their model back on the training data that it already seen and also on the testing data, which was the 10% that they held back. And again, they evaluated using this mean squared error and R squared here. So you can see in the training data, it was very good. So it has an R squared of 99%. In the testing data, it was slightly worse, but still very good at sort of 98%. Obviously, there's not a huge amount of data in this test data set, but still it looks like as a concept, it works pretty well. And they also plotted for the 10 different sequences in the test data, just the target versus the predicted. And in this case, their target was associated with some level of error because of the sort of experimental way that it was calculated. So it's just showing the error against the prediction as well then. So overall, their model seemed to work pretty well with their data. So that's cool. We're going to have a look at doing it ourselves. So we'll go back to tidy models, which again, I sort of introduced last time, but essentially it provides a consistent interface so that you can prepare your data, construct your models, and evaluate your results in a consistent way. And it means you can easily move between different modeling packages without having to change your data and your code in a load of different ways. So it makes it much more user-friendly to try different model types and things. And as a reminder, the kind of code structure that we have with tidy models is we will firstly create a model. not on any particular data, just the type of model and the settings that we want to use. We'll prepare our data, so do any filtering or scaling or pre-processing that we need to do, split off our training and our testing data, or cross-validation. And then we train the model, so we bring together our data and our model and define the variable to predict. And finally, we can test and use the model on new data after that. So to do this support vector machine in tiny modules, we can use an engine called KernLab. So this is the package in the background that is actually running the model and training it and everything, but Tidy Models lets us use it without having to learn all of the specific details of it. This has our 3 tunable parameters that we had seen before. So we have our cost, our sigma, and our margin, which were the 3 we talked about earlier. Up here, I've inputted the values from the paper as well. This time we also have to set a mode, which we didn't last time. This is because support vector machines can do either classification or regression, so we have to say which one we want. So in this case, we're doing regression. Pre-processing requirements for this are we need factor or categorical predictors to be converted to numeric values. We saw that earlier. And our predictors should have the same scale. This isn't an issue here, but this is just making sure you don't have really high chunk of values and a really low chunk of values for different predictors, essentially. So the new thing we're going to introduce with this paper is automation with recipes and workflows. So because we're doing slightly more complicated pre-processing this time, we're going to convert our sequencing data to dummy numeric variables. We're going to use these recipes and workflows. So recipes let us specify our pre-processing steps. And then it will apply all of our pre-processing steps to any data that we give it going forwards. So we can apply it all to our training, to our testing when we use that, and also any future data that we might want to run through our model. So this allows us to avoid any manual pre-processing, which can be tedious and also error-prone. So it stops you from accidentally doing something slightly differently or in a slightly different order that might give you slightly different results. So it makes your models more reproducible if you use these recipes. And then we can use a workflow to bring everything together. So it brings together our recipe and our model and lets everything be run at the same time. So the steps for this, we're going to create a recipe. So we'll tell it our formula and show it our data, although it won't actually input it yet. So this is the same as we saw with the model. And then we can add pre-processing steps to it. So this is where we'll set our dummy variables. Then we can create a model in the same way as we did before and create a workflow to combine all of this. Okay, so given that, we'll go over to our studio and look at some code I prepared earlier. So the first thing we're going to do is set up the packages that we need. So we're going to use tidyverse and tidy models, which is what I've been talking about. Everything else is just sort of aesthetics for this, and this just means that any functions that are in tidy models, it will use preferentially to any functions that have the same name in any other package. So we'll run that, and it will load in our libraries. Slightly slowly. Then the next thing we can do is read in our data. So this is our sequencing data, and if we have a look at this, you can see we've got IDs for different promoter sequences that we have. We have a strength value for each of these, and then we have for each position in our promoter what base is there. So we've got lots of G's in position 1, A's and a G in position 2, etc. And for this, there are 226 columns, so there are 224 bases across our sequence. So the first thing I'm going to do is split our data into test and training data. I've done this based on what they said was in their testing data, so I've just pulled out those IDs. You can also do this randomly, but I thought I'd keep it similar. So if we look at this, this is our training data. We've got 90 sequences in here, and this is our test data. We actually only have 9 in here because there's one that's missing, but we will not worry about that for now. So then the next thing we're going to do is define our model. So as we saw, we're going to use this SVM RBF. I'm using the same values as they used in the paper, just for this example. The engine that we need is kernlab, and we're doing a regression, so we'll set that. So if we run this and have a look at it, we can see it tells us we're doing a radial basis function support vector machine model, which is a regression model, and it has put in our main arguments for the cost, sigma, and the margin there. We can also look at the translation, which is what it will be given to this kernlab package using this translate function here. So if I run that, you can see it tells us, again, our model at the top, but it also tells us the kernlab package function is KSVM. It has some missing arguments because we haven't actually given it our data yet. And it's put in our cost, epsilon, which is our sigma, oh no, sorry, epsilon, which is our margin, sorry, and sigma, which is our sigma. And it's also picked a kernel, which is RBF, which is what we want. So the next thing we're going to do is create a recipe. So this is going to pre-process our data. So to do this, we can use this recipe function here. We want to predict strength. So we're doing strength given dot is everything else. So we're using everything else in our data to predict the strength of our promoter. And we pass our data in here just so that it can see the structure of it. It doesn't actually use the data at this point. Beyond that, the first command here I have is update role. So before, I think I just removed the ID column before adding it into the model, because you don't actually want to predict based on the ID. It's different, obviously, for each sample. But when you use this recipe, you can set a role to tell it that your ID column is an ID column, and therefore it won't treat it as a predictor. It will just be a label. The next thing we're using is this step novel. And we're doing this for all of the predictors in our data. So we're now ignoring the ID because that's no longer a predictor. And this essentially adds a novel factor for things that are not seen in the training data. So if at any particular position in the sequence there is a base in the test data that was not seen in the training data, it will add another sort of column for something new that it hasn't seen before, and will mean that our model won't shout at us for having things that it's never seen before. It will just go into a new category. The next thing we're going to do is step dummy. So this is going to do what I showed you in the slide, where it will create binary columns to convert our bases into a numeric value. And finally, I'm using this step underscore ZV function, which removes predictors with 0 variance. So if at any point there are no t's, it will remove all of the t's and ignore them, because it's not going to contribute to our model in any way, because there's no differences between any different strength of the promoter there. Also, just to say, this step dummy that I'm using here obviously can be used for many different categorical variables. I'm just using this one as an example of using it, but it doesn't have to be applied to like this kind of sequencing data. In fact, it's probably better if you have more different categorical variables. Yeah, so I'm going to run this and we can have a look at our model here, or our recipe, sorry, not our model. You can see the input. We have a number of variables. So we have our outcome, which is 1. So that will be the strength. We then have 224 predictors and an ID column. And it tells us the operations that it's going to do. So we're going to do a novel factor. We're going to do dummy variables for all of our predictors, and we're going to do a 0 variance filter. So that's useful. What's also useful is to have a little look at what our data would look like once it's run through this recipe. So to do that, we can use this prep function, which will just run your actual data through your recipe and let you have a look at it. And we can pull out this thing called template here, which will just show us what our data actually looks like. So here we've still got our ID column, we've got our strength column, but now we have a whole load of other columns that are position 1G. So this is whether or not there is a G at position 1. You can see there are a lot of them. This one here is not a G, it is something else. Obviously, at this position, there was only G's and one other base that it could possibly be because we only have one of these. So we don't actually know which other base it is from this because we've compared it to a baseline rather than comparing or rather than having a column for everything that's been seen. And we can see that across all of our different positions and different bases, we have a different set of combinations of zeros and ones along here. So that's what it actually looks like that we're putting into our model. The next thing we can do is create a workflow that combines our recipe and our model. So we can create a workflow here. We then add our model using add model and call our model that we made earlier. And we add recipe, which is again the recipe we just made. So if we run that and have a look at it, you can see it's a workflow. We have a preprocessor and a model. Our preprocessor has 3 recipe steps and our model is down here, so we can again see all of our main arguments for this as well. So finally, we can fit our model. I've set a seed here because there is a random element, and so it will mean that we will get the same model back every single time. So to train our model, we use again this fit command here, and we give it, in this case, just our training data because we just want to train it on the training data and not on all of the data. So if we run that, it will fit our model, and then we can have a look at it. So if we just look at our fit model, you can see this is now a workflow that has been trained. We still have our recipe and we still have our model, but we have some more information in our model too. So we've got the same parameters we had before with our epsilon, cost, and sigma, and it also tells us the number of support vectors that we have, which is quite interesting. The last 2 bits here are essentially evaluating how good the model is. So training error, as you might expect, smaller is better. Objective function value is essentially to do with the margins, and on the whole, larger is better for that. But we won't worry too much about the details of those. So then we can pull together some predictions. So we can run our, or we can pass our training data to our fitted workflow now, rather than just a model, to predict it. So that means we don't have to do any pre-processing to our training or our test data. It will automatically run through the whole recipe and then predict based on that. So we can do that on both the training and the test just to have a look at it. So we can see that here and we can plot it to make it slightly better or slightly easier to visualize. So this here is our training data. You can see there is almost a perfect fit between our prediction and our strength. This is our testing data. Doesn't look so good. So probably here we have overfitted our data given how we have approached this, which is slightly different to how they did in the paper, which is probably why theirs gave much better results. That being said, something that's really nice about having used recipes and workflows is that we can really easily change certain parameters and things within our workflow without having to redo everything. So say we see this and we're like, this isn't very good. I want to try doing a polynomial support vector machine instead. Then we can do that very easily. So if we just define a new model, for a polynomial SVM, we can use SVM poly this time. I set a degree equals 3. So this is looking at original squared and cubic essentially. We're still using the same engine and the same mode because we still want a regression. But we're just changing that to be polynomial rather than RBF. So if we run that, All the rest of the parameters I'm just leaving on default for now, just for simplicity. And then we can update our workflow with our new model. So we don't have to redefine our recipe, we don't have to redo anything else. We can just take our original workflow, pass it update model to it, and provide our new model, and then we will get a new workflow with instead a polynomial model. So we can run that and fit a new model based on that. And if we have a look at this, we can see we now have a polynomial kernel function rather than our RBF function that we had before. You can see we have slightly fewer support vectors, only 86 in this case. And again, we can have a look at this so we can predict our training and our testing data, and we can, again, just plot it down here. So we can see on our training data, we get a slightly worse fit, so it's maybe slightly less good on our training data. It also, in this case, looks fairly rubbish on our testing data. But again, in this case, we haven't optimized any of the parameters in any way. I'm just showing this as an example of how we would run it, rather than fitting a particularly good model where we've cross-validated and picked the optimized parameters. Okay, so I'll head back to my slides just to show you some examples of some other parameters and things that we might use, different model types, just as a comparison. So in the training data, we can see these are the paper parameters. They're probably a bit much for what we've done here. You can see the number of support vectors on this was 90. And it really overfit our training data. It's pretty much perfect correlation between them. Just changing some of the parameters here to the sort of defaults in the RBF, you can see we now don't fit our training data so well. We have fewer support vectors. And we can also have a look at linear and polynomial. So this is what we looked at just now. If we look at linear, we can see this actually looks pretty similar to the polynomial in terms of how well it fits on the training data. This has the fewest numbers of support vectors. But if we look at the test data, we can see, again, we haven't optimized any of these parameters, so this isn't super surprising that the more complicated kind of models don't do so well. If we optimized and made the model a better fit for our data and how we've approached this, then we would probably get some better results. But based on not really optimizing, actually the simplest model has given us essentially the best response here. So we have, or the best prediction here. So we have an R-squared of 0.67, which is a lot lower than in the paper, but still a lot better than this 0.042 that we saw using their parameters in our little example data there. So what they then did with their model, they did some further exploration where they looked at the impact of single base mutations on predicted promoter strength. So they would change each base in the promoter sequence to a different base and run it through the model and see what the model predicted the promoter strength to be. So you can see here this change at position 20 from A to G greatly reduced promoter strength. We have a super strong one down here, which was an A to G at position 209 that had a really strong increase in promoter strength as predicted by our model. So you could use this and potentially test some of these and see how accurate the prediction is on different mutations that weren't necessarily included. They also compared to a previous approach. So they had done a previous paper where they'd looked at a similar thing, but using neural networks rather than SVM. They thought SVM might be better because it works, or it can work better on smaller data sets than neural networks might. But even using these different model types, when looking at this kind of summarization, the vast majority were consistent between the two. So the same mutations were either increasing or decreasing promoter strength. And only about 20% of the different mutations had sort of absolute differences in these predicted values more than sort of 0.2. This is also just showing the predicted versus the target strength in the training and test for the support vector machine in blue and the neural network in red. So the SVM does seem to work slightly better for them in this case than the neural network. So just a reminder that automatically going for the most complicated model, which would be the sort of neural networks, which I haven't really discussed in these series, but we will come back to possibly next time or the time after. But using a more complex model is not always inherently the best choice. Sometimes using a slightly simpler or less computationally intense model can give you the same or even slightly better results. So just a reminder, some sessions coming up. So the next one we'll be looking at classification. We're actually going to keep looking at sequences because this was a super simple example of how we might deal with sequences, but there are actually potentially much better approaches in how we can pre-process our sequencing data. This was also a very easy example because it was looking at one specific promoter where all of the sequences are the same length. But if we have more complicated sequences and they're of different lengths and things, then that can cause issues when we're trying to pre-process to get a particular model input. So we will look at potentially better and more complex approaches to including sequences in our model outputs. As model inputs, next time. And then we'll look at a couple of other examples, so looking at interpretable models in the fourth one, and looking at some feature extraction in imaging data, which is a really big area in machine learning in the final session in August. So this is just a reminder of the dates as I showed you on the previous screen. So with that, I will wrap up and thank you for listening.